causes global warming, and we can use this basic science knowledge to improve our life, to ensure our survival, actually. Or, for example, learning that fishes can regenerate most of their parts, like their heart, can get us knowledge to learn how we can improve our life quality or life expectancy, right? So we must promote basic science research. Wherever we are, if we are the government, if we are researchers, we must promote it, right? And this is how we do it in Ecuador. Given all this lack of resources or whatever we might have as a barrier, we must do it, right? And in Ecuador, we do it first by inspiring people with our own research, and then inspiring people through education, which is what I'm gonna talk about. Um, she's Eugenia Del Pino. She single-handedly founded a developmental laboratory lab in Ecuador 40 years ago. She established the developmental biology class, so we teach to undergrads, and she has built a research unit only based on undergraduate students that are able to publish, that are able to generate first class research by being very creative with the resources we have. She's laureate in a lot of ways. She's the most important researcher in the country, and this is the way she has established and inspired research in Ecuador by her own research. I took this picture when I joined the lab like 12 years ago, and this is when I left the lab to go to my doctoral appointment and to my PhD appointment, and now I'm back. I assume I'm the head of the lab now, but and she's retiring, but she's still mentoring us. Um, so what she did as a researcher is understanding frog development. So that's what we've done. So most frogs develop like in a very fashionable way. So they lay eggs outside, they divide, the cells divide, they then gastrulate. So gastrulation is a very important process because it creates it ordinates the embryonic layers outside the ectoderm that will form the skin, the neural system. It makes the endoderm to go inside. That makes the digestive tube, and the mesoderm that forms the muscle and the bones that gets in the middle, right? So the gastrulation is a group of movements in the embryo that makes the layers to go in order, right? To make a living thing to orderly, right? And once the layers are in order, the embryo knows how to make a head, a tail, a body. It makes a nervous system here labeled. It living organism. We've learned that by studying these alternative systems like frogs. And then in frogs, of course, they form this tadpole that goes and swims outside, right? Um, and we learn that by studying frog development. However, we also found some frogs that don't like to develop in this way, right? This is a very interesting frog that when it lays the eggs, the mom pushes the eggs back to a pouch that it's on the back, and the embryos actually develop in the back of the frog. So it's some kind of bizarre parental care. Um, and these frogs, of course, don't develop like most of the frogs. So they have these, they get into gastrulation. However, they don't form the initial intestine we call archenteron. They don't form it, and they actually form this weird disc of cells from which the whole embryo develops. All right, so it's an embryonic disc like most of the amniote animals, reptiles or mammals or birds, but this is a frog, right? So it's a very weird and unique frog, and Dr. Del Pino, she based her research on studying why this frog develops like this. So we started to look in every frog we could get our hands on, according to the diversity we have in Ecuador, right? And we actually found that there's frogs that delayed their developmental movements, their developmental processes. They can do it, right? So, for example, this is the model frog for embryo development, Xenopus, and they form the mesoderm normally, here labeled by the notochord, the most axial, the most dorsal structure, um, and they form it really fast. Other frogs, like this dendrobarid frog or the, Gastrotheca here, they can take their time. They delay the formation of the mesoderm. They actually separate the formation of the head mesoderm from the notochord. Notochord. They can delay it and separate the structures, right? So we found an evidence of modular development just by studying diversity of amphibians. And this delay in processes is mainly related to the adaptations, the reproductive adaptations they have, right? So these frogs don't have parental care, they just leave the eggs there, they have to develop fast. So all the developmental processes occur fast. These frogs, they have parental care, they take care of the eggs that they leave in the ground. This frog puts the eggs on their pouch in the back so they can delay and change the processes accordingly, right? So, and you can see all the adaptations that, for example, mammals have under development. We can find all such diversity in vertebrates. We find it in just the studying frogs, right? So here is where I started. I joined the lab 3 months ago, and I'm gonna show you a little bit of what we are doing now, mainly because we are passionate about finding the evolutionary keys from the origin of the vertebrate pattern, because we can find them all studying frogs, right? Um, so this is one of my thesis students now. I remember I have to remember show you that all these students are undergraduate students. Most of them came with these ideas to me, and I'm fostering those ideas. I'm trying to make them uh, publishable. I'm working with them to make them nice. So once we know all this developmental diversity, we want to go into the evolution of these patterns, right? So this Jennifer is sampling all the clade of dendrobarid frogs, right? And we found some very nice differences. Like they have huge difference in the size of the eggs that they lay, huge differences in the timing of development. They differ a lot in the way they form the gills, the embryonic gills. However, they still do a similar gastrulation. So this is what it's called an evolutionary sand clock, right? So there are patterns that are conserved across evolution. There are patterns that are divergent that depending on the species. So she's working on this really nice question now, the genetic and evolutionary basis using the dendrobatic frogs. Um, this is Anna Belen. She actually found this really nice frog that apparently don't need to form a blastocell. It's an embryonic cavity where all the gastrulation movements occur. Apparently, this frog don't need that cavity to develop normally. So we're learning how and why this frog does that. Um, she's Maria Jose, and she's a really nice herpetologist, and she is curious about the pigmentation in the eggs, right? So she is studying these glass frogs, so the skin is transparent. Studying these frogs, she actually found that some frogs, some species have pigment in their eggs, some species don't have, right? And apparently that depends on where they lay the eggs. So the ones that are pigmented are in contact with the light, the sunlight. The ones that don't have pigment apparently are hidden in the leaves. However, we want to know the genetic basis of this, and she's been going to the field a lot, and she actually found a species that actually lays both kind of eggs, so pigmented and not pigmented. So we are doing genetic experiments with this to find, of course, the genetic basis of this. Um, these two guys are working on a reptile development, so we were able to find a stable population of those snakes that they are reproducing, and we're analyzing some the way the face of the snake develops, and the way the yolk has evolved to provide nourishment to vertebrates from the frog to the reptiles and then to the mammals that have lost it actually, right? So these guys are interested in that. Um, and then I'm gonna summarize really quickly what I did in my PhD research in the US. So we work with frogs. Now I want to introduce the model of zebrafish to use it in the lab for teaching and to use it for research, right? So this is a larva of zebrafish, and here is a transgenic line that is labeling the neuromast. So the neuromast are sensory organs in fish that are used to sense water movement, right? They have sensory cells. They are called hair cells, and these hair cells are homologous to the inner ear hair cells, right? The only difference is that the only difference is that fish can regenerate these sensory hair cells while mammals cannot regenerate these sensory hair cells, right? And the advantage of the zebrafish is that we can look at in vivo, right? We have transgenic lines, we have tools, a lot of tools to follow regeneration of sensory cells in vivo in the zebrafish. So I want to use that. Um, this is a slide that actually shows my 6 years of PhD research. And the thing I did is found some molecular cues that guide the regeneration process that show it. So there's actual molecules, there's signals that can be manipulated that show to this cell regenerate and form a new sensory cell, or to say to the epithelium to recover to form more stem cells to sustain the epithelium. That's what I found in my PhD research. And the interesting thing is that all the things that I found were done by using chemical inhibitors, chemical manipulators, chemical drugs that can to manipulate these pathways. For example, the notch pathway, they can be manipulated by using drugs, right? So if you guys remember, I mentioned that our country is very rich in diversity, but most of the times we don't know what to do with that diversity. So right now the government is promoting a lot of research in bioprospection, so to find active molecules in the environment, in this diversity we have, to find active molecules with the chances of being used in the industry, of being used in medicine, etc. So what I'm trying to do, what I'm looking for funding, what I'm doing now is using these biologically active compounds that are being actively researched in our country to analyze regeneration in the zebrafish lateral line. As you have seen, there's multiple projects going on. Some of them are basic science. Some of them are translational science that we can easily do in our country, right? The other part is to inspire people through education. And this is something that also Dr. Del Pino started 40 years ago. We are continuing it, we are improving it. So this is the only class of developmental biology that is being taught in the country right now. We're trying to fix that. But of course, we are starting with our own university. So we have a class that it's 3 hours a week, theory and practice. In theory, we analyze current events on research, such as regeneration, stem cells, modern techniques like transgenesis, gene silencing, bioethics, things that sadly we can still not do practically. However, we have a really nice practice class and field trips on which we were able to teach, to inspire students to learn about development, about embryonic development, by using sea urchin, frog development, zebrafish, chick development. We actually scan the field for eggs, for developing organisms that we can find in the forest, and we analyze them in situ. We bring the lab to the field. We work with them. This is one of the places we did. It's a really nice place where we collect sea urchins. We in the field we are able to work with in vitro fertilization. We work with that with the students. These are all pictures taken by the students. They were able to do fluorescence staining, to analyze development, to stage the development of the sea urchin by working in the field. So this is basically what they do. This is the way we inspire people to follow a career, not necessarily in developmental biology, but in basic science, right? With that, I would like to thank the lab. This is the first picture of my lab. I hope I have one every year. I'd like to thank the Universidad Católica. They are the main source of funding in my lab. Right now, it's the only one. I would like to thank a lot to Dr. Del Pino for their mentoring, for the inspiring research and life. Hugo Navarrete and Mercedes, they are the heads of the department who has been very supportive to me. And of course, the students, which are the inspiration for me to continue with these projects. Thank you very much.
Thank you, Andrea, for your excellent talk. And let's move on. Our next speaker is Dr. Miguel de Carvalho, and from the Pontifícia Universidade Católica do Chile. So again, another Pontifícia Universidade Católica. But before the beginning of your talk, it's our great pleasure and honor to announce that Dr. Miguel de Carvalho was a recipient of a prize from the TUAS. So he received a prize for his work on statistics, and so we have to please applaud him for this. And here is his diploma from the TUAS, Third World Academy of Science in Rolac, and also the check. So congratulations. So let me give you good luck. So you have to sign here.
Thank you very much.
So we have to take a picture. My opinion, he received a prize on statistics, but so far all the people that speak here in this stage would also deserve a prize. So congratulations all the 4 morning speakers, as well as the 2 that we have already listened to. So let's continue with our Dr. Miguel.
Well,
Anything else that we should do?
Well, thank you very much. This really now gets me even more nervous. And actually, I start here with explaining the why the 17 here and not the 18 as it is. I actually took very serious the preliminary program where it was written before that it would be the 7 meeting. So take home message: don't prepare talks in advance. Okay, so my talk will be about some statistical and probabilistic aspects about extreme events. Well, they're typically known as well as black swan events. And what's the idea here? So suppose you have a random sample of losses, and think about these losses as losses associated. Well, it can be losses associated with a certain portfolio, but it could also be losses associated, say, with the consequences of something like an earthquake, for instance. Okay, So suppose we have a random sample of losses. And now suppose that we would like to say something about what's the probability of observing a loss which is largest than the largest one we've observed so far. Okay? Coming back to the example of the earthquake. So, for instance, in Chile, earthquakes are recurrent, and they're definitely cause lots of losses. Now, we would like to say something about the probability of observing an earthquake, for instance, which is largest than the one ever registered. Okay? Now, if you use standard statistical methods, you run into troubles. Particularly if you use the distribution function, the empirical distribution function, and this would be an undergraduate course in statistics, formal, okay? That probability would be 0. So what can you do? So what can you do? So actually, as put simply by Nassim Taleb, the fool believes that the tallest mountain in the world will be equal to the tallest one he has observed. And somehow it reflects well the fact that this probability here of observing something which goes beyond your maximum is 0. Okay? So this is the starting point. We need methods which need to extrapolate, but the extrapolation is into the tail of the distribution. So the mass that is left there that we're not accounting for. Okay? Why do we care about this? Well, because we care about extreme events. So what are extreme events? So extreme events are events such as the floods, recent floods in Australia, well, earthquakes in New Zealand, Japan, Chile, Hurricane Katrina itself, mudslides in Rio de Janeiro, or 11/9 World Trade Center. Okay? So we want in this field of statistics, which is statistics of extremes, we're typically concerned about modeling small probabilities, but underlying these small probabilities is a rare event which is catastrophic. That's typically the idea, okay? Okay, so we don't work with the normal distribution. If you're working with sums, you have the normal distribution as a consequence of the central limit theorem. Here, the law of interest is the law for the standardized maxima. So we think about maxima here. And the distribution of interest is the so-called generalized extreme value distribution. Okay, which is a distribution which has been widely studied. Now, what happens when you want to consider the extremes of more than one variable? Because in some cases you will want to focus on those. For instance, in some atmospheric problems, you are not just interested in understanding what's the probability of observing an extreme level of a certain contaminant, but of 2. And the combination of 2 at the same time may be catastrophic. If you're regarding the topic from a finance point of view, and if you have a portfolio, say, with just 2 assets, then if you think about the losses on one asset and the losses on the other asset, you don't want to get losses which are extreme simultaneously, because that will mean a huge loss for your portfolio. Okay? Okay, so this distribution here will no longer work, because we also need to model the dependence between the extremes. Okay? Think about Pearson correlation. Think about correlation. Now we need correlation, but now for the extremes. Okay? Ideas such as correlation, but now for the extremes. Okay? Now the problem is that when we want to model that, okay, we still have a distribution, but it is very challenging to fit that distribution. It is the so-called bivariate extreme value distribution, which is a distribution which has this form here. Now, where this parameter here, the parameter which is describing the dependence between the extremes of random variables, it's a parameter which is a probability measure. So how can you do that? How can you fit this extra parameter? And not only that, this needs to be defined on the 0-1 interval, and it needs to have mean 1/2. Okay, that's the analog of your Pearson correlation. So you want to assess the dependence between the extremes of two random variables. Okay, so essentially, well, both start with exponential. That's true. So now things here become much more challenging because now the parameter here of interest is, as I mentioned earlier, also a distribution function. And so how can we learn about this object? Okay, remember that the idea was extrapolation, to say something beyond the observed data. Remember about the black swan event type of idea. Okay. So this talk is centered around three articles. So the main ideas are on 3 articles. One which has been recently submitted, a book chapter which will appear in 2016, and an article which appeared on 2014. So let's move to the most exciting part. Okay. So far, what have we regarded? We've regarded ideas about, okay, it's important to learn about the extremes, because the extremes are typically associated with ideas of risk. Okay, and also about, okay, dependence now becomes interesting, but at the extreme levels of two random variables. Okay, so what if that extremal dependence is also changing according to a predictor, in the same way that we use regressors in linear regression? What if now our parameter of interest, which is extremal dependence, is also changing over a predictor, a covariate, or say over time? How can we model that? Okay, so we need an object which allows us to describe dependence between the extremes, but now over this predictor. So we need more than a distribution function. We need a family of distribution functions, which is indexed here by x, which is the covariate of interest. Okay, so the idea is similar to linear regression. In linear regression is statistics 101. Okay, so the basic principles are how can we incorporate this dynamic into the extremal dependence structure, okay, into the dependence between the extremes. How can we allow for the dependence between the extremes to change over a predictor? So essentially, we want to move from this, which would be the object, which would be telling us there's lots of dependence here between the extremes of these two variables. But now we would like to get some dynamic to this object. We would like the profiles of this surface to correspond to things like this. Okay? And the more mass you have close to 1/1/2, it means the more dependent are the extremes of your random variables. Whereas more mass close to 0 or 1, such as in this case here, it would mean higher level of extremal independence. Okay, so your extremes should become independent. So from the point of view of what I was mentioning in terms of the interpretations in finance, so if you have loss 1, loss 2, higher dependence between the extremes, it's really bad, because it is as if you were losing twice. It means you're not diversifying risk. Okay, so Whereas if you have more mass here close to 0 or to 1, it means that it's okay. You're diversifying because the extremes of your random variables are not as correlated in the extremes as in the other setting. Okay? So whereas in correlation, you think about perfect correlation, correlation equal to 1. Now here the object of interest for describing correlation between the extremes is this H, and the more mass you have here close to 1/2, the more dependence you have between your extremes. Okay? I'm trying to make some connections with statistics 101. Okay? How can we estimate this H? Okay, now there's the challenge because this H needs, it can be virtually anything, but it needs to be on the 0-1 interval. And for each value of X, for each value of your predictor, this needs to have mean 1/2. Remember, we mentioned that here right from the start. So there's a theoretical condition that tells, okay, but H can be anything which is defined on 0-1 interval, but it needs to have mean 1/2. Okay, so here is an estimator for H. So this is a smooth estimator. You smooth on two directions, on the direction of your predictor or covariate X, and you also smooth on the direction of W. Remember that there's zero direction in terms of X and the direction in terms of W. So, and you take these convex linear combinations of these beta densities here. Okay? So essentially these are weights which are based on a kernel K, and this parameter takes this form here. Okay? I'll open it a bit more here, because I want to move to the application. Okay, so I mean this is simple, very simple to implement, and it will obey the moment constraint. Okay, so for each value of X, you will always have mean equal to 1/2. Okay, so this would be a valid estimator for the spectral surface I mentioned earlier. Okay, so there's a few parameters here involved. I'm skipping the details because I want to move to the application. Okay, so now how can we apply this? Think that we would like to model the dependence between the extremes, the extreme losses in two markets. So you have two markets of interest, and you are concerned with the losses on these two markets. Okay, and so we have data from our sample period covers these years 1988 to 2014. So it covers the last economic crisis. So, and we'll focus on losses. Okay, let's have a look at the data, because I think it helps. Okay, so let's focus here on, I'll focus here on CAC DAX. Okay, and what we have here at CAC DAX. So CAC is the French market, DAX is the German market. Well, it's the indices corresponding to this market. And you have here what? You have here a time varying color palette for these scatter plots. So if you look here, so gray points essentially mean more recent observations, which took place more recently. So you start seeing here that there's quite a bit of gray points, quite a bit of gray points here at this zone. Okay, which would mean simultaneous losses on both variables. Okay, let's zoom this. So if we zoom this zone here, what we get is the following. Okay, so well, there's definitely here a few blue points, which would mean points which took place in the earlier 90s, but there's really lots of concentration here of points here which are related with more recent events. So that could suggest what? It could suggest that the dependence between the extreme losses on these markets would be what? It would be increasing. That's what this time varying scatter plots are actually suggesting. Okay, what's the corresponding spectral surface telling us? It definitely verifies also these as well. So as you keep moving over time, you have what? You have more recently, you're more mass close to 1/2, which is a symptom of higher level of extremal dependence, which means that losses in one of these markets are occurring at the same time more often than in the past. Okay, so joint losses are happening more often than in the past. Whereas here at the beginning of the observation period, you actually have your more mass close to 0 and 1, which is a symptom of asymptotic independence at extremes. Okay, so what if we just fix here a few periods of time? Okay, some related with some events of interest. Say a decision by the European Central Bank or so. Well, we can take any profile that we would like here, any cross section of the spectral surface. And for instance, this one is on 2010. So something like this. Okay, so we fix here 2010 and we take that slice. And this is the uncertainty evaluated through a bootstrap. So definitely we see here more dependence at the extremes recently, okay? And let me, I have 2 minutes, so let me just summarize. So, I mean, the methods that I've been working recently are for modeling what I call non-stationary extremal dependence structures. So essentially, these are related with ideas on conditional risk modeling. Okay, so you think about risk, but from a conditional perspective. Okay, and essentially, what we can learn also from the data, well, we applied this to this setting, but there's other settings where I believe this could make sense. So the setting that we applied was on a finance case study where we have assessed the dynamics governing extremal dependence of a few markets. Okay. And so, well, the last thing I think I would like to say, well, would be obrigado. Okay.
Thank you very much, Miguel, for your talk. And now we put my glasses. Now we have time for discussion, and I would like to call the 3 speakers here in front, and then okay, and then we can direct the questions to them. And okay, so raise your hands if you have anything to ask.
I'd like to take the opportunity to have all these young people here, just to remind, have you understood what happened this afternoon here? Did you really understood? Did you understand? I mean, for example, we have everything about science here, okay? One thing is science driven by passion. From one side, and the other side is driven by utility. This is the word we live today, how to do science. Most of us is driven by passion. This is what we do. Okay, this is our first option. We want to do science, we want to know things. And for example, André, it's your name, isn't it? André gave us a lesson of passion. You know, he had a mentor, a lady that I know so much because she was always in the TWAS meetings, and she's fantastic. She's always pushing us to talk something about something. and you did it quite well. I think that you showed us how passion can move science to the future. Know things, understand how nature is, how nature is working. From the other side, Cecilia came here, and when I read the title of your lecture, I suppose finally we will listen something telling us what means innovation, because innovation is in our mouth for the last 20 years at least. And we do not know how to do it. Okay? And I want to question you one thing. For example, do you believe as the things that you brought from Mexico? Yeah? I should say that this is not different what we have in Chile, it's not different that we have in Argentina, in Brazil, in Peru, Colombia, all the and other developing countries. They are fighting like crazy to be innovative, to do something to sell to the others, okay? But we are doing in one direction, as you said, is the top government and everyone saying what we should do. Do you understand that? What we are doing, we listen that we need to innovate. Yeah? Have someone sought to do other way round, to start from the bottom. And if we do not change the country, if you do not have the courage of challenging, there is no innovation. Innovation is not appearing by law, I mean. You know, and teaching people. You do not teach. There is something that appears, you know, innovation. And when we are trying to say, we need to do innovation, there is a very big discussion in Brazil. Is innovation a challenge for university, for enterprises, for them together, for them combining something? And I think that we need first to teach people, everyone in a country, that behind a money card, there is a lot of science and technology. Behind any telephone that we are using, there is a lot of science and technology. If we do not know why we have to believe in science, if everyone in the street, in Brazil, everyone is all the time like this, you know, everywhere you are going, there is someone with a telephone. Doesn't matter if they are rich or if they are really poor, they have a telephone. But do they know that there is a lot of science behind it? How to innovate if they do not know? It's something that they take for granted. It appears in the world, you know. The telephone appear, the airplane appear, the frigo appeared in the world. There is no science and technology. Okay, I don't want to be very long, but just to show to young people that we live in these two worlds today. We still have space for doing science for passion, or we have to do science only for utility, that someone will use it. This is a very huge discussion in several countries, mainly in the poor countries, because we do not have resources to support both kinds, you know? And I think that is a very, very good opportunity for you young people to think about. And the third point is Miguel. Miguel is someone that got a prize. It's a winner of a prize. I will not say no. It is representing something that for people doing science is a huge thing to get. You know, I think that all people here would like to have 1 hour in the field that you choose to work. And mainly, if we do like you do with passion, you just transmit us here passion. Not, I do not understand what you've been talking about. I did not understand. No, it was. I'm trying to understand. I know what swan is, black swan also, but I would like to do one question for you. Is it possible, with your formula, to help Brazil go out of the crisis that we we need to take advantage of the science that we are learning here, because this is, you were talking about the European market. Maybe it's good for you, but not for me. I don't care about the European market. I'm talking about Brazilian market. And I think that, and Andrea, I think that you gave a good lesson here, because normally we are not looking to our environment, that we have a lot of things to learn if we are just looking, this is science, just to see what's happening, why you have all these animals. And I think that you are lucky to live in Ecuador. This is a fantastic land with so many things that is preserved, what is not happening in the other parts of the world. Okay, three of you, my congratulations. It was a science talk with all the limits, Thank you.
You did a fantastic summary, I guess, about my feelings. But so, is there any other questions? Otherwise, I will pass the word to them.
Yeah.
So,
just about what you say, I guess the way would be to teach students curiosity, and I think that's a very difficult thing. Right now, I'm teaching biology to people that are gonna be nutritionists and developmental biology, and in both, I'm experimenting by making them create hypotheses of anything they are interested in. And I mean, ideas come, right? Ideas come very fast. A lot of people, I know a guy that wants to develop this kind of food to make people that work in the circus like work faster, like a nutritional supplement to people that works in the circus, right? And another guy, I haven't teach them this, and he came out with this brilliant idea of why the blastocyst don't develop in frogs, right? And so ideas come, you have to nourish them for entrepreneurship, and if you nourish curiosity in any level, I guess you will get to ultimate economical benefit, but most importantly, to get curious people all the time. Yep.
you want to comment something, because there is another question there, but maybe
yes,
you can let her make her comments and then we'll pass the word to you.
Okay, I just want to tell that these both sides that you told, passion and also the utility, are not in the opposite ways. I think that they are supplementary, because I am a passionate person that believes in the utility of research. So the point is that right now it is for the region very real that we are in a situation, in an economic situation that could change at any time. Almost all of us depends on funding, public funding, most of us. So sometimes it is not previsible that this funding will be stopped, because the first thing that governments stop is culture, science, and technology. So if we are dependent on that, which would happen with researchers, the most close example is Spain. So they are unemployed right now, most of the researchers. So we have to think about some alternative ways to provide our own funding. So this could be from the industry. The problem is that the intercommunication among researchers and the industry, because we perform research only thinking about humankind and to learn more, and because of our passion, but not really because of the necessities or requirements of the industry. So this could be an alternative way to provide some questions and to access to some funding. Also, the point is that young talent people have several ideas creative ideas. Most of them were shown by Andre, that showed that all the research that he performed in his laboratory comes from the ideas of young people. So why not monetizing them? It is a possibility, and a great necessity is also to overcome the technology dependence of our countries. So if we have talent, if we have infrastructure, if we have almost all the elements, I think it is a possibility to reach this goal. So that's it.
Okay, so go ahead.
Yeah, I have a question for Andres, maybe Cecilia as well. I've known several cases where let's say developed countries actually do a lot of research in Mexico, in Chile, and in Ecuador. But actually, how much of that is actually how much of that stays in Ecuador, for example? There are many groups that are actually doing plenty of research, right? In Galapagos, for example, and many companies as well go there, or not Galapagos, but in Ecuador, and actually for a lot of money. How is the community getting something back from that? I mean, you talk about society, you talk about investment, but not every investment means company. Sometimes means something for the society, which could be education, which could be knowledge, right? What's going on in those cases?
Yeah, I mean, I like, that's what I probably personally I like basic science, because it's far away from the profitable point, but it's really close to the use of it, right? So the knowledge about our diversity stays with us, right? We researchers in ecology, in evolution, in developmental biology, we hold that knowledge for us, so it doesn't run away with like the money. It's not patentable at that point, right? And that's what I like ecology and evolution. We sadly, we as a small country, still a developing country, we depend from the research in applied science, the applied research, we depend from outside, basically. And the idea is not depend on it, so to generate our own knowledge, to fix our own requirements, right? Our own necessities, like hunger, like the main causes of death in children or in productive population. Those things, we still don't have them. So hopefully in the future, if we get people interested, we will get our own research for our own people instead of going outside.
Any further question? So I just want, okay, sorry,
If I just have a follow-up on the, I think also in terms of the ideas of innovation that we were discussing, I think it would be also interesting to hear some thoughts also about, I mean, many times I work mostly in teams of statisticians, for instance, a few times as well with medical doctors, economists as well, but innovation should also come, this is a question, should it come also from more interdisciplinary crosstalks, if you will? I mean, and sometimes the language is definitely a barrier, I feel. I mean, it seems like each science is speaking like its own jargon. And I mean, when we go back to Karl Popper said something which was very, very interesting. There's really no disciplines, there's just knowledge. And why are we having then different languages if we just have a common knowledge? So if you like to have a follow-up on this.
I think that what we are doing nowadays is just to be appropriate about questions, so that we have human questions. And of course, it's not a matter of one person, it's everybody. If we are talking about cancer, we've been listening to here today. Now, who is the god of cancer? All of us, because we need to think about it. We go from sociology of cancer to the therapeutic
epidemiology.
So we need to have everyone looking. If we're talking about diabetes, a very huge problem nowadays. Who is taking care of diabetes? Everyone, even the man of the supermarket, is taking care of diabetes. And violence, water, energy. I think that we need to think about the questions, not only to have patents, but to solve our human questions that we are very slowly in solving this question. Probably, if we work all together, probably we can find an answer in a quicker time. That's the idea.
Yeah, integral solutions.
Yeah, we are nowadays we are using the word convergence of sciences. So to not say multidisciplinary or transdisciplinary, so it's common words, because every time you insist on talking about disciplinarity, you are thinking
compartment.
Convergence is better. No one knows who is there. Converging everyone.
So if you don't have any further questions, so thanks again our three wonderful speakers.
Thank you.
And it's not written in the program, but we do have a coffee break, 10 minutes. It was not programmed, but we decided to have it. So just have a small break to refresh and to have some caffeine. Thank you very much for attending this
I think a lot of money and this is a different thing. What you write
Desafia muito, né? Quebra todos os paradigmas. Então você pode dizer assim, tá, agora vem que ela vai chegar. Não vai ter tempo de pensar em mais nada. Bonito. Esse juntar de trabalho o público faz mais até onde eu tenho direito. Isso é que só o Deus do mercado.
Mesmo sem dinheiro. Aí você pega 10 reais e dá
One more paper. Yes, one more
Very
paper.
small.
One more paper.
E aí eu dou uma conferida, aí quando eu pego essa lista, acaba que você está falando,
Só não pode ter gente, não pode ter lá pessoas que nem todo mundo ali é falar. Mas se o nome não estiver aqui, tem que a não ser a Adriana não vai. Pode não. Uma pessoa não
Okay, so good afternoon. So my name is Flavia Gomes. I'm from the Federal University of Rio. So first of all, I'd like to thank you for staying so far. So this section will have only two people. Unfortunately, the third one had some personal problems, so he couldn't come. So you're gonna probably finish a little bit earlier. And first, also I'd like to thank the Brazilian Academy of Science and also Vivaldo, I don't know exactly where he is, and Deborah, for giving me the opportunity to take part in this event. And I really think it's a great opportunity to put people with different backgrounds, different expertise, as we have been discussing from the beginning of the day. And I really believe that's the way we should put science forward, putting together people with different way of thinking and to work. And so I really hope you enjoyed this meeting. So that's one of the main motivation we had when we tried to organize the program. So today I will have the pleasure to be the chair of the section on genetics and evolution. So for that, I'd like to invite Dr. Francisco Cubillos from Universidad de Santiago de Chile, and he's going to talk about understanding the role of the intergenic regions shaping natural phenotypic variation is. So thank you very much for coming. It's a great pleasure. We'll have 20 minutes and then.
Yeah. Thank you for the introduction. Today I'm going to talk about what is my passion. My passion is genetics, ecology, and evolution. So in the beginning, I'm going to show what we do with genetics. And then at the end of the talk, I'm going to show how do we sell these genetics. Because as we've been talking today, people ask, so what you do, what is it good for? I mean, how am I going to use what you're doing? And in particular, funding agencies usually ask, so what's the application of what you're doing and how can this impact society? I'm a geneticist. I like genes. I like DNA. I don't care where, I just like DNA. So at the moment I work in a food research center, so I have to focus my genetics in things that affect food. So we work with food safety in particular and different food products. In Chile, we export a lot of fruits, a lot of wine. So we really worry about that. And in particular, my research has been focusing in mycotoxins. So mycotoxins are toxins produced by fungi. Usually you have this beautiful plant, this like, it could be maize. And if you, and if this product, this fruit see rain, then you have these fungi, happy fungi that are going to grow here and are going to contaminate this food. If you have tons of these, you're not going to see them actually, and then you're going to feed chicken or cows, and then you're going to eat that meat, and then those toxins are going to get to you. And usually these toxins can cause directly cancer, right? You can also find mycotoxins in beer, in wine, and everything that is derived from any plant, okay? Because there is humidity everywhere, and there is fungus everywhere. So we like to use biocontrollers for that. We like to use the solution that nature has created. We don't need chemicals. We believe that there are already some organisms that can control these. And at the same time, we are a wine country, as you may know, in Chile. So we work with yeast, and yeast is able to work as a biocontroller of these toxins, and at the same time is able to do the fermentation process, right? Beer, bread, wine, etc. And at the same time, yeast is an excellent model organism for doing genetics. So I've been doing genetics and trying to apply it to these processes so I can sell the genetic science. So we work in particular in genetics in what is called complex traits. What is a complex trait? It's a trait that is complex, like a height, human height, that's complex. And when I mean complex, it means there are several genes affecting the trait, all right? I mean, we're not tall or short, we're not black or white, right? We can range and we can vary from different colors, from different heights, from different weights. And this is because there are several genes controlling a trait. Cancer is the same. You're not sick because you have a single gene that causes cancer. you're sick because you have a combination of genes. And to understand that, we use a bit of mathematics. I mean, I don't use mathematics, but I talk to mathematicians and I tell them, well, you know, I have a trait and I know that this trait is controlled by genes, all right? And I know which genes are those, or I have mapped, I have found some of the genes. However, these genes don't act on their own. They interact with themselves, so it depends on the genetic background. It depends what genes you have, what genes gave you your mother, your father, and so on. And at the same time, they interact with the environment. One thing is to be susceptible to cancer, for example, lung cancer. Another thing is to smoke, right? If you smoke and you're susceptible, you're likely to develop cancer. However, if you have the genes, but you don't provide the environment, it's very unlikely that it's going to happen. And at the same time, so the interactions. And I've had some hard time trying to collaborate with mathematicians and statisticians. I'm actually trying to collaborate with a Susana collaborator, and in the same university, in Pontificia Universidad Católica de Chile. And I've been doing some genetics lessons to her, so we can actually, you know, try to collaborate and try to do something interdisciplinary, right? So we like to study this, how genes behave and how the genes behave in different environments in order to predict and in order to understand different traits, right? So in the future, we say, well, we can have the genotype of your son, and based on that, we can predict, right, some of the phenotypes, how likely is that he's going to have a disease, for example, which is very scary. So the question is, where is variation coming from? And so we have a phenotype, right? I'm going to focus on yeast now. So yeast do fermentation, and when you do fermentation, for example, you produce ethanol, right? And you can produce different levels of ethanol. So there are different types of yeast, just like us. There are yeast from different types of from different parts of the world, and they're going to differ on their DNA sequence, right? But at the same time, they can differ on the RNA. The RNA is what comes from the DNA when it's expressed, all right? And then the RNA can be converted into different protein and different metabolites. So if we have differences at this level, then everything downstream can change as well, right? And this is going to cause overall that we are different. That's why we're all different from each other in this room. And if we look from the mathematical point of view, to explain different levels of metabolites in different individuals is more difficult than to explain differences at the DNA level, all right? So as it goes down, it's more difficult just because you have more factors influencing each one of these points. So in the lab, we're interested on this, on the RNA. So the DNA, the gene is going to express, and it's going to express on different levels, and this is what we want to understand, and how different RNA levels affect a phenotype. So I'm going to introduce our model organism to study genetics, and it's budding yeast. So this is the organism, Saccharomyces cerevisiae, is the one that does the wine fermentation process, but also lives in the wild, like in oak trees, and it could be a pathogen if it has the opportunity. So there is a lot of natural variation, even though it's the same species, it can live in different places. Also, there is the beer fermentation. It has a very short cell cycle. In 2 hours, you can double the population. So if you have 1 million cells, 2 hours later, you're going to have 2 million cells. And also, it's got a small genome size. So it's like 100 times, the genome is 100 times smaller than the human genome, all right? So it's got a lot less genes, and it's much simpler to try to study and to understand. So in approximately in the year maybe 2006, 2005, people from the Human Genome Project in the UK was trying to sequence 1,000 different individuals, human individuals, all right? But first, before going into human, they wanted to do it in a simple organism. So they go in touch with the yeast community and they ask, could you provide us strains and isolates from different parts of the world? So this small map here shows where the isolates were obtained from. So there are some from South America, some from North America, from Africa, from Europe, from Asia, and Australia. So just like us, yeast have been in our pockets forever. Probably the Spanish were the ones that brought the yeast from Europe to America, and those are the ones that are doing the wine at the moment around here. So they have kind of followed our own history.
So this is what they go about the structure of the strains. This is how they behave. Imagine these as different individuals. The closer they are, the more similar genetically they are. So what these guys found is that there were some groups, defined groups, that were actually quite different from the rest. And if you see them, there are some from North America, some of them are from Asia, Africa, Japan, and this is a one European cluster. So these strains are either isolated from vineyards or from Europe. This is sake, so it was isolated in Japan from sake fermentation. So these are genetically quite different, and actually represent quite well what the human distribution is. And at the trait level, they're actually quite different. So what you do in yeast, you put the yeast to grow in an environment where you provide one carbon source. So for example, you provide sugar, and then you measure how much they grow on this sugar, and then you're going to see differences. And some of them like one sugar more than the other. Some may like fructose, some may like glucose, some may like maltose. It depends where they come from. The ones that fermentate beer like maltose because it's the predominant type of sugar. The ones that are in wine, they like much glucose. And at the same time, you can try and see different inhibitors, right? In the environment, they see many growth inhibitors, and therefore they have adapted for that, all right? So phenotypically, they're quite diverse as well. So they represent a very good model to study genetics and complex traits. So how do you study complex traits? In a way that you cannot do in humans, or it wouldn't be ethical to do it. You take two different individuals and you cross them, and then you generate an hybrid. So imagine that this is your mom, this is your dad, then you're the hybrid, and from the hybrid you can get a progeny, right? And this progeny is going to be a recombinant from those parents, right? If the parents are one from Asia, for example, and the other one from Africa, these are going to be recombinants. And the good thing about in yeast is that you can generate like millions and millions of segregants. So you can have different combinations of genes that you're not going to have in any other way. So we can do what is called genotyping. So we know what the genes are and what the alleles are throughout the genome for all the different individuals. And at the same time, we can do phenotyping. So in yeast, what we do, we grow them in different conditions. So this is what we call heat. We grow them at very high temperature, and then we see which strain grows and which one doesn't. And when you do this in the individuals, then you can try to correlate. So if these are markers, you can say, okay, so we take all those that have, let's say, imagine that this is the African parent and this is the Asian parent. Let's say it doesn't matter if it's got the African or the Asian gene, they grow the same, or the growth rate is the same. However, if you look at another marker, you can see that one genotype grows less than the other genotype. So you can say, okay, it's the Asian genotype that allows them to grow at high temperatures, right? So then you can assign the function of that gene and of that allele in particular. And at the same time, at the ecological point of view, you can say this explains the adaptation to an environment. And if you do this genome-wise, so these are all the chromosomes in the genome, then you can say where all these peaks represent different genomes for where you've seen adaptation, and then you can say in this part of the genome, you can assign that is the responsible for the variation in the population, in this recombinant population. So in humans, most of these peaks are not within genes, but actually are within these regions that are between genes. If you think, maybe you've seen in the news or you read somewhere 5 years ago, 10 years ago, when they started to understand the human genome, they said, well, there's a lot of regions that don't code for anything. They seem to be just junk, right? It seem to be like rubbish. They don't do anything. But actually what they do, they control the expression of the genes. And 90% of the differences between us in this room are located in these regions. They're not in the genes themselves. They're not differences and mutations in the genes, but actually are differences within these regions. So we got interested on analyzing these regions in yeast because it's possible to do it more easily than in humans. And for this, what we do is imagine that this is a mutation that affects a gene. This is called open reading frame, which means gene, right? And we have this regulatory region. So we have a polymorphism or a mutation here that affects a gene. And then we can use the polymorphism within the gene as a marker, as a phenotype, to see what this polymorphism is doing on the expression levels. So we do an hybrid. We take two different individuals, again, like an Asian and an African individual. We do an hybrid, and then in the hybrid, we measure the expression of this allele and this other allele, the two versions of the gene. So we've used our 4 favorite strains representative of different parts of the world in yeast. We did crosses very easily. We did this in triplicates. As we increase the number of replicates, we reduce the noise of our measurements. And so we grew them in very rich media. They grew very happily. Then we did what is called RNA sequencing. So this is like the genomics era. So reviewers and people that publishes papers really like this. So you get data for the whole genome. And then we can estimate for a single gene how much expression you have from one allele and the other. This means if I take your genome, I can measure how much your maternal and paternal allele expresses. So I can say, okay, for this gene in particular, the allele that your mom gave you expresses more than what your dad gave you, all right? And we can do this genome-wide. So we've been trying to correlate this with some phenotypes. The yeast genome has 6,000 genes. We were able to evaluate 3,000 genes after doing some filtering. And we did this for the 6 crosses. So we had 4 strains, 6 crosses. And in some crosses, we found about 30% of the genes had differences on the levels that those 2 alleles behave. In some other crosses, we had up to 60% of the genes in the genome behave differently. This means that for the same gene, the 2 alleles that your mom and your dad gave you behave in a different way, right? In humans, it's not different from that. And this is what you see like in a plot. The red dots are all those genes that are differentially expressed, or those alleles are differentially expressed, and the black dots are those that are being equally expressed within an individual. This is a cross between a North American and a Japanese strain. So these results suggest that actually what this is called allele-specific expression is commonly found in yeast and allow us to determine then what is the consequence of having different expression levels. If I have a mutation in this region between genes, what is the consequence, all right, at the phenotypic level? Phenotype seen as a trait, all right, as we look. So for this, and this is when I started selling my science, I had all these results and I had to do something more applied. So I work in a wine lab, or I join a lab where we do a lot of wine, and now I started to make some beer, which is not very nice in the lab, actually. You just keep buying what you buy nowadays. I'm not there yet. But what we've done is we've obtained different alleles. So we said, okay, we're going to try to apply this to wine research. And we selected some alleles from the one European strain that seemed to be quite different from all the rest, right? One of the strains was obtained from a vineyard, all right, from grapes. So we wanted to understand. And we found some genes. This graph represents this kind of orange graph represents expression levels of the European strain, and you can see that in all these genes is higher than all the rest. So it could mean something for the fermentation process. In particular, this gene hasn't been much studied. All these other genes are known to participate, and they've been published to participate in the fermentation process, but this gene wasn't very well known. So we thought this is quite interesting and actually could serve for the industry. This gene is, if it's being highly expressed, maybe it's doing something for the wine and it could be used. So we did something quite sophisticated. I'm not going to go into details here, but what we did is we took the region from one strain and copied that into the other strain to know what it was doing, right? So we took that, put it in the other strain, and then we know if it actually goes up or to lower levels. And we did this for the promoter, which is a regulatory region, and then we did this for the gene. We wanted to know if the regulatory region or the gene were responsible for the differences that we were observing between these two strains. So we made wine in the lab. This is called synthetic wine must. We made wine in the lab. We had the North American strain, which lives in an oak tree, clearly doesn't like wine environment. And we took the wine strain, we did the fermentation, and we evaluated for ASN1, which it takes asparagine and aspartic acid and convert it into asparagine. What was going on with these two amino acids, because nitrogen is essential in the fermentation process. So the one European strain it's got 100% relative production, and here where you can see the North American strain, when we change either the promoter region or the gene itself, we can see that there is an increase in the consumption levels of this amino acid, which is the one European strain really like this amino acid, right? Because it's not present in high amounts in the grapes, so it has adapted to be quite efficient eating this amino acid and assimilating this amino acid, but the North American strain is not. But when we put these one European regions in the North American backgrounds, we can see that the levels of assimilation increase, which means that actually these two regions are responsible for the variation. So here where we see is that we're mapping the mutations, and the regions are responsible for this. And then we found that it's because of a transcription factor in the regulatory region that binds to the one European strain and not the North American strain that could explain the differences. So to summarize, we've seen that expression differences are highly widespread in yeast. We've seen that looking at this cluster of strains and genes with related function, we can actually find some alleles of interest. And we have found what could explain the phenotypic differences between the strain, and try to make something applied, or try to explain what can we do with our research. This is my, so far, a small team that I work. This is Mara, a master student. Cindy and Valentina are two technicians that are working with me. They are biotechnologists, and she's veterinary. Felipe, who is also a biotechnologist, he's been doing the beer, she's been doing the wine, and these girls have been doing all the molecular biology. Also collaborating with different people, because I believe that through collaborations we can actually do science. And the funding agency, because they've been finding quite a lot of my ideas, and I'm very thankful for that.
Francisco, thank you very much for the enthusiastic presentation. So let's move to the next speaker. So I have the pleasure to call Dr. Jennifer Alcaino, also from the Universidad de Chile. And the title, I think I'm going to leave for her. I think it's too difficult for me. And okay, so thank you.
Thank you, Flavia, for your presentation, and I would like to thank the organizing committee for your invitation. It's been a pleasure for me to be here. So, well, today I will talk to you about the genetic control and regulation of the biosynthesis of isoprenoids in the red yeast Xanthophyllum isodendrose. Isoprenoids is one of the largest family of natural products, where more than 50,000 different structures have been described. These molecules are involved in several biological functions. Here I just included a few. And our group of work is currently interested in sterols, which are important structural and regulatory components of eukaryotic cell membranes, and also in carotenoids that are natural pigments of yellow, orange, red color that are involved in the protection against the photooxidative damage. Carotenoids are also very important from a biotechnological point of view. It is estimated that the global market of this pigment will reach 1.4 billion by the year 2018. And this market is led by beta-carotene, followed by lutein, and in third place by astaxanthin, that is the carotenoid that we work with. Astaxanthin is best known for its use in aquaculture, where it gives the characteristic color of the flesh of salmon fish, which is required by the consumer. And also it is used in the cosmetic companies because it has potent antioxidant properties. The yeast Xanthophyllum isodendrose is one of the few natural sources of astaxanthin. Here in this picture you can appreciate the color of the colonies of this yeast, which is due to the accumulation of this carotenoid. And carotenogenesis per se has been described in this yeast And one particular point that is characteristic in the carotenogenesis of Xanthophyllomyces dendroros is that a cytochrome P450 enzyme is involved in the synthesis of astaxanthin, unlike other astaxanthin-producing organisms, which I will refer only as CYP in the next of my presentation. Well, this is the isoprene molecule of 5 carbon atoms, which is the building block of isoprenoid compounds. And different subunits of isoprene give rise to the wide diversity of isoprenoid compounds. The active form of isoprene is isopentenyl pyrophosphate and its isomer dimethyl allyl pyrophosphate that are joined together to give geranyl pyrophosphate, and the addition of a second and a third molecule of isopentenyl pyrophosphate gives farnesyl pyrophosphate and geranyl pyrophosphate. These 3 reactions are catalyzed by prenyl transferase enzyme, giving the precursor of monoterpene, sesquiterpenes and sterols, and also of diterpenes. Geranyl pyrophosphate is also the precursor of carotenoids, as the union of 2 molecules give rise to phytoN, which is the first carotenoid that is synthesized in carotenogenesis. So, as I told you in the beginning of my presentation, carotenogenesis per se is known in Xanthophyllomyces dendroros. However, little is known about the early steps of carotenogenesis. So one of the first questions that we wanted to address was, which are the prenyl transferase enzymes that are involved in the synthesis of geranyl pyrophosphate? To the time, it was suggested that only one prenyl transferase was involved in this 3 steps. However, in literature, we found that it was possible that 2 prenyl transferases could be involved, following this first model, where the first one, encoded by the FPS gene, catalyzes the first 2 reactions, while the second one catalyzes the third reaction. So we analyzed the genome of this yeast, and we were able to find 2 potential prenyl transferase encoding genes that we named FPS and CRT-E, and we expressed these genes in E. coli in order to perform prenyl transferase enzymatic assays, using the protein extract of the transformant bacteria, and also using labeled isopentenyl pyrophosphate with carbon 14 and a variable substrate, which could be dimethyl allyl pyrophosphate, geranyl pyrophosphate, or farnesyl pyrophosphate. So when we expressed the FPS gene, we were only able to detect prenyl transferase activity when using the first 2 variable substrates in addition to isopentenyl pyrophosphate. However, when we expressed the second gene that we identified, we didn't find prenyl transferase activity with these 2 substrates. However, we did find with farnesyl pyrophosphate. So with these results, we were able to conclude 2 things. First of all, indeed, the 2 genes that we identify encode prenyl transferase enzymes, and also in Xanthophyllomyces dendroros, there are 2 prenyl transferases involved in the synthesis of geranyl pyrophosphate. So we were able to contribute a little bit more about the knowledge of carotenogenesis in this yeast. As I told you in the beginning of my presentation, there is a CYP enzyme involved in the synthesis of astaxanthin from beta-carotene in this yeast. And CYP enzyme, or cytochrome P450, it's a large family of M-containing proteins that catalyze the hydroxylation of the substrate, as you can see here in this general reaction, in the presence of oxygen and NADPH. And to perform this reaction, these proteins require a redox partner, as the electrons are not transferred directly from NADPH to the enzyme, but through this redox partner. And in class 2, cytochrome P450 systems that are anchored to the endoplasmatic reticulum membrane, the general redox partner is a cytochrome P450 reductase, or CPR. So, if in the synthesis of astaxanthin there is a CYP enzyme involved, there should be a redox partner also involved. So we look in the genome for a potential redox partner, and we find a gene that we name as CRTR in order to keep the carotenogenetic gene nomenclature. We mutated this gene, and you can see at the naked eye difference in the phenotype of the strain, where the first one is red and the second one is yellow. We extracted total carotenoids from both strains in order to to confirm our visual observation, and we separate them by reverse phase HPLC, and we were able to distinguish a major peak in the wild-type strain that corresponds to astaxanthin. However, we confirm that the mutant strain does not produce astaxanthin, and it accumulates another carotenoid that corresponds to beta-carotene. So, in conclusion, this gene is essential for the synthesis of astaxanthin in Xanthophyllomyces dendrorose. This is a very interesting point because in an organism, there can be many CYP encoding genes and CYPs that catalyze different reactions. However, in most cases, there's a single CPR encoding gene. So we ask, what happens with the CYP-CPOM, the complement of cytochrome P450 in Xanthophyllomyces dendrorose, and we analyzed the genome, and we were able to detect 13 potential CYP encoding genes. We still don't know the function of all of them, but among these genes, 2 of them share high percentages of identity and similitude with CYP that has been described to be involved in sterol biosynthesis, which are CYP61 and CYP51. So, as I told you at the beginning, sterol are important components of eukaryotic cell membranes, and ergosterol is the main sterol produced by fungi and yeast, including yeast, which is equivalent to cholesterol in mammalian cells. So, we ask ourselves, if there is a CPR that is involved in the synthesis of astaxanthin, what happens to the synthesis of ergosterol in this mutant strain? As also CYP enzymes are involved. And also what is interesting, that sterols as carotenoid are isoprenoid compounds, so they derive from the same precursors, as you can see here. So, we did extract total sterol from both strains, and in the wild-type strain, we were able to identify a single sterol that corresponded to ergosterol as expected. However, in the mutant strain, we were able to identify to distinguish 3 different sterols, where the one in the middle corresponds to ergosterol. So, with this, we were able to conclude that indeed, the synthesis of sterols was also affected in the mutant strain. However, this mutant strain is still able to produce ergosterol, suggesting that in this yeast there are alternative redox partners that can sustain the synthesis of ergosterol, but not the synthesis of astaxanthin. So, in order to prove the functionality of the CYP encoding genes that we identify, we mutated them. We begin mutating CYP61 gene, which should be involved in one of the last steps of the synthesis of ergosterol, and we confirmed that this mutant didn't produce ergosterol. But for our surprise, we observed that all the mutants that we create from different wild-type strains share a similar phenotype, which is a more intense red color than the wild-type strain from where they derive. For example, this is a mutant that derives from this strain. So, in order to confirm that there was a change in the composition or quantity of carotenoids, we extracted carotenoids of all these wild-type strains and mutant strains. Here are the results. And what I want to highlight, that indeed, carotenoid production is enhanced in the mutant strains in relation to the wild-type strain. So, in the absence of ergosterol, because of the CYP61 gene mutation, this branch is enhanced, synthesizing more carotenoids and, of course, astaxanthin, which is the end product of this pathway. So, even though the early steps of carotenogenesis are not still well known in Xanthophyllomyces dendrorose, it is known that isopentenyl pyrophosphate in non-photosynthetic organisms like yeast are synthesized through the mevalonate pathway. This pathway has not been formally described in our model yeast. However, we have identified all the potential genes that could be involved in this pathway. And among them, I highlight this gene, which is HMGR. There it goes. that encodes the hydroxymethylglutaryl-CoA reductase. And what is important about this gene, has been characterized in other organisms as an important point or a bottleneck in the mevalonate pathway. And its expression is regulated at different levels, for example, in mammals, where it produces cholesterol. So, in order to understand a little bit more the phenotype of our mutant, we analyze the expression of this gene by RT-qPCR. And we observe, this is the growth curve of both strains, which is quite similar. We analyze the expression at the stationary phase of growth, and we observe that the expression of this gene, at least at transcript level, was almost 20 times higher in the mutant strain. So this could be one of the reasons why carotenogenesis is enhanced in this strain, as the bottleneck gene is being overexpressed in these mutant strains. We did analyze also the other genes involved in the mevalonate pathway, and we observed a similar pattern, a higher expression in the mutant strain. So, this is what happens with the other genes involved in the mevalonate pathway, which their expression is also higher in the mutant strain. So, our current question is, how does ergosterol regulate its own synthesis by affecting the mevalonate pathway? And we have a candidate gene, which is SREBP, for sterol regulatory element binding protein. This protein is involved in a complex regulatory pathway that has been extensively described in mammalian cells, where cholesterol plays a fundamental role. However, in fungi, little is known about it in fungi, and it has been studied only in Schizosaccharomyces fombe as a model yeast, and also in Cryptococcus neoformans, but nothing is known about other fungi. So, in very simple terms, what happened with this pathway? The SREBP protein, it's a transcriptional activator which is synthesized in an inactive form, as it is anchored to the endoplasmatic reticulum membrane, where it interacts with another protein that it's able to send sterols. So, when sterols levels are sufficient, this complex stays at the ER membrane. However, when sterol levels are reduced, this complex transfers to the Golgi apparatus, where the SREBP protein is proteolytically cleaved by proteases, and the N-terminal domain of this protein is released and can travel to the nucleus, where it dimerizes and activate the transcription of target genes that include an SRE sequence, sterol regulatory element sequence, in their promoter region. And in fungi, among the target genes in fungi, there are the genes involved in the ergosterol biosynthesis and also in the hypoxic response. So, considering the phenotype and the experimental evidence that we have about this mutant that is not able to produce ergosterol, we think that this pathway is also present and operative in Xanthophyllomyces dendrorose, and in this case, as this strain doesn't produce ergosterol, sterol levels are reduced, then the SREBP is always on, is always active, activating the transcription of genes involved in the mevalonate pathway and also in carotenogenesis, resulting in a carotenoid overproducing phenotype. So, in order to test our hypothesis, we first analyzed the promoter region of several genes involved in the mevalonate pathway, carotenogenesis, and ergosterol biosynthesis from Xanthophyllomyces dendrorose. In the ones that I highlight here in red, we were able to find potential SRE sequence. But also, we have experimental evidence of these genes in blue that might be regulated also by this pathway in our yeast. So, we align the SRE sequence that we identify to get the consensus sequence of the SRE element of Xanthophyllomyces dendrorose which is this. And this is the one, oh, I'm sorry, but it moved configured the picture. What I wanted to show is that it's quite similar to the one that has been reported for Schizosaccharomyces pombe. So, moreover, we have also identified a potential gene that could encode the SREBP protein. And the deduced polypeptide from this gene contains all the structural elements from the SREBP protein family, which are a serine-rich and a proline-rich region at its N-terminal domain. Also, two transmembrane segments that would allow this protein to anchor to the ER membrane, and most importantly, a basic helix-loop-helix leucine zipper motif that would allow this protein to dimerize and to bind to DNA. And also, in this motif, we align it with other SREBPs. We were also able to detect a conserved tyrosine, which is only present in SREBP proteins, but not in other basic helix-loop-helix containing DNA binding proteins. So, in order to prove the functionality of the gene that we identify, we mutated this gene, and when we culture it in YM-rich medium, we didn't find any phenotypical difference with the wild-type strain. However, what is interesting, when we culture this mutant in the presence of cobalt chloride or acylic compounds like clotrimazole and ketoconazole, which are antifungal compounds that specifically target the ergosterol biosynthesis, the mutant is not able to grow, unlike the wild-type strain, suggesting the role of this gene, as I explained it in the model before. But what was most interesting, when we mutated this gene in a non-ergosterol producing environment, which is the CYP61 mutant strain that doesn't produce ergosterol, we saw that this strain also is not able to grow in the presence of this compound, but most importantly, we observed that the carotenoid levels were reduced, reaching again the wild type level production of carotenoids. So it seems that indeed we have identified an SREBP encoding gene in the genome of this yeast and prove its functionality. So in conclusion, we are studying the whole SREBP pathway in this gene, in this yeast, and we have been able to identify several other elements, and we're working on them, and we think that this pathway could be an important target to be manipulated in order to favor the synthesis of carotenoids in Xanthophyllomyces dendrorose. So, thank you very much for your attention, and finally, I would like to thank Fondesit for giving us the funding to perform our research, and all the genetics lab team that work with me. My colleagues, Victor Cifuente and Marcelo Baeza, the technical support given by Salvador Barahona and Dionisia Sepúlveda, and all the students that work with the three of us. Thank you very much.
Thank you. Thank you very much. So I think now I'd like to call. So now we have time for the discussion. No? Are you tired?
Tired.
Yeah.
Thank you. I wonder, how do you discard spontaneous mutations? I think if your yeast is a clonal population or is an heterogeneous population.
Can you hear? Is it working?
Ahí. No, parece que tiene que estar ahí, ahí, aló. Ahí, tiene que estar apagado.
I can use one.
That's a very good question because we face that quite often. Yes, these are current populations. If you keep the yeast for about a couple of months growing, like just changing to rich media every time, then you start getting some spontaneous mutations. But these strains, they have been originally sequenced, like when they were, well, in the picture that I show, when they were sequenced, we know how they are. And then if we do another experiment and if we see something, like we always control for a spontaneous mutation, if we see something that is not in the original sequence, then we know that it's a spontaneous mutation. Usually spontaneous mutations arise when you apply selective pressure. Let's say we want to see a resistance to high temperature, and then you see that a clone is going to rise quite quickly just because it got a spontaneous mutation within a gene or somewhere in a regulatory region, and it's going to take over the whole population. Yeah. But at the same time, yeast is quite good because sometimes you have different clones within a single population. Let's say you have 1 million cells, and those 1 million cells are not going to be the same. And that's what happens with tumors, for example. You can have several hundreds of cells, and they don't all behave the same. I mean, usually cancer cells are because of mutations, and they're not going to necessarily have the same genotype, and they're going to behave differently as well.
Did you were able to characterize the changes in the regulatory regions you found? And if are you planning to do some sort of evolutionary study of how did they lose the changes or got the changes for?
Yeah, so I didn't have much time, but we mapped the region that we think is causing these differences. And this is one of the genes that we found, but we have several other genes, and this is for wine. But my main focus now is mycotoxins because I really care about public health. So yes, I have some ideas about how to use experimental evolution to understand how the different genetic backgrounds behave when they face a toxin. The question is whether some genetic backgrounds resist better when a toxin, when they get contaminated by a toxin. Let's say if from us in this room, some people would be more resistant to those toxins than others just because of the genotype, the genes that they have. So that's an experiment that we can do in yeast, and at the moment we are doing it. Yeah, good question.
So my question is to Jennifer. I'd like to hear your explanation for the data, the protection of all these compounds compared to the yeast cells. So are these protections related to oxygen express that protects again these compounds, these three compounds that you tried? And also about if you can play with genetics and envision like biofuel production. Biofuel, yes.
Yeah. The last three compounds that we analyzed, cobalt chloride, yeah, still we don't know what is the action of this compound, but it has been reported in literature that the cells respond in the same way as when exposed to low levels of oxygen. So it's a mimicking agent of hypoxic conditions, but the biological response is still not known well yet. And the other two compounds, which are azolic compounds, these are the classical antifungals that we have available, that the target is the ergosterol biosynthesis, specifically at the first CYP enzyme, which is CYP51. So these agents target this enzyme, and this reduces the ergosterol biosynthesis at a very high level of the synthesis of sterols. And by doing this, other sterols are accumulated, which are toxic to the cell, and the cell dies. And then you asked me another thing about biofuel. Yeah. In general, we are just starting to study this pathway, the SREBP pathway, but it has been mostly studied because of its potential to be used as a therapeutical target for antifungal for fungal infections. However, with our results, we have seen that it regulates the overall synthesis of isoprenoids. So Xanthophyllum dendrorose is a very interesting model because we can easily study at least two of them, which are sterols and carotenoids, what has been doing. But personally, I think to manipulate this pathway could help to make an isoprenoid cell factory, if it's always active.
So, Francisco, I do have a general question about the role of coding regions in evolution. And so there's some evidence that few cells, several cells from the same individuals might be genomically completely different. So I was wondering if you have any evidence or feeling or idea what's the impact of non-coding regions for the cellular diversity of a human being. For example, there are millions of different cell types in our nervous system, or 100, and only a few cell types in the heart. Right. So is there any?
Yes. So,
did you get the question? I mean,
yes. So usually that's called epigenetics. So well, you have that the cells are going to behave differently just because they're going to be, for example, the regulatory regions could be methylated or not. And that's going to imply that that gene is going to be active or it's not going to be active. So if you look within a single healthy individual, usually that's the mechanism how it controls. Plus, you know, the trans factors and all the transcription factors that are going to control how that tissue is going to behave. Then correlating that a bit with the natural variation, and what I was answering in that case, in non-healthy individuals, then you can have subcells or populations that have acquired mutations, and those could be within either like within a gene, for example, or within regulatory regions. Mostly regulatory regions are account for the differences that are within us in this room. There are not bad mutations. Usually they are sustained in the population. That's why they're there. They're neutral. They don't generate big changes, but there usually are small changes that all together, you know, all the mutations together generate big changes, but each of them individually, they generate kind of small changes. Then you have different behaviors if you look at cell types, and then if you have like diseases and so on, it depends. But usually regulatory mutations are the ones responsible for differences between our cells, actually.
More questions? Okay, so I think that we should thank you again. And Vivaldo, I don't know if you want to give any advice for tonight or tomorrow or. Oh. Okay.
7 p.m. the bus. And tomorrow morning.
Fogo do chão.
There is no beach tomorrow morning.
So I hope to see you tomorrow at 8:30. Not 9, 8:30. Okay, thank you.